Hi everyone, welcome to your lecture on dominance relationships. So last lecture we talked in depth about genotypes and phenotypes. So given the example right here in the picture, what is your genotype? What are the genotypes? And what are the phenotypes? The genotypes are your homozygous dominant, heterozygous, or homozygous recessive, represented by the letters that you'd find on your genes. And the phenotypes are how it shows up. Purple and white flower color. If you show the dominant phenotype, what could your genotype be? If you look here, you'll see that all three of these purple flowers, there are two different possibilities. They can either be homozygous dominant or they can be heterozygous. So you are not sure. If you see the dominant phenotype, you are not sure what the genotype is without further testing. If you show the recessive phenotype, you know that it must be homozygous recessive. So if you want to find out what the dominant trait's genotype is, you're going to do something called a test cross. And this is going to help you to determine the genotype of the original dominant organism. So you're going to take an individual that has the dominant phenotype and breed it with an individual with the recessive phenotype. So you take, in this case, we're using purple and white flower color. So you take a purple flower and breed it with a white flower. You know that the white flower is homozygous recessive. If 100% of the offspring in the first generation are dominant, then that means your original was purebred. So that means if all of the kids are purple, if you breed a purple flower and a white flower, you get a bunch of kids and all of them are purple, then your first purple flower was homozygous dominant. That means that all the kids are then heterozygous. If the offspring are not 100% dominant, if you have some white kids in there, then your original purple flower must have been heterozygous. It must have had a little allele or the recessive trait to pass on to the kids in order for some of them to show as white. And you can prove that with a Punnett square. This is what it looks like with a Punnett square. So when we use Punnett square, they're usually good for only a single trait at a time, and they have just 4 boxes. When we look at just one trait at a time, it's called a monohybrid cross, mono meaning one. This is an example of a Punnett square that we would use if we wanted to look at 2 traits at the exact same time, and it's called a dihybrid cross, and it has 16 squares now. So we're going to try a couple of these in class, a couple dihybrid crosses, but we are looking at 2 different traits. We're looking at seed color here, either yellow or green, and we're looking at seed shape also, either wrinkled or round, and they don't really have anything to do with each other. So we're going to do a dihybrid cross, and I want you to write it all down in your notes. So we're going to first show the probabilities of a cross between 2 people that are both heterozygous for full lips and a cleft chin. Full lips is going to be represented with F, the letter F, and a cleft chin is going to be represented with the letter C. If they're both heterozygous, then you know that full lips is the dominant trait, cleft chin is the dominant trait, because that's what they're showing. Those are their phenotypes. And heterozygous means they have one of the dominant and one of the recessive traits, so they have a capital and a lowercase of each. So their genotype is big F, big C, little c. So full lips would be dominant to recessive, or to thin lips, which is recessive, and cleft chin would be dominant to non-cleft chin, which is recessive. And if you guys aren't sure what a cleft chin is, it's when you've got that little dimple in your chin. Sometimes people call it a butt chin. So if you don't have that, then that is going to be considered the recessive trait. If you do have that dimple in your chin, then that is going to be considered the dominant trait in this example. So we're going to do the hardest part first, because the hardest part is segregating the alleles. Remember that that just means to separate them. So if you have one parent that's big F, big C, little c, and another parent that's big F, big C, little c, you're going to do every possible combination of an allele from a full lips allele with a cleft chin allele. Let me show you what that looks like. You do a technique called foiling in math, first, outer, inner, last, and we're going to use that same technique to separate these alleles. So we'll start with one parent first. So big F, big C, little c. You'll do the first F with the first C. So that's big F, big C, and put that down. Because both parents are the same, you have one parent that has the same stuff here and the same here, you can put the same information. If the parents are not the same, then you're gonna have to FOIL them separately. So one big F, one big C. The next one, you have a big F with a little c, the outer F with the outer C. Big F, little c. You can write that also over here on the side. Then you have the inner F, which is little, with the big C. Now remember when I had said before that the dominant version always goes in front of the recessive version? I was talking about for the same trait. So these are two different traits. So we're going to have the lip trait before the chin trait, always. The lip trait before the chin trait in this example. We're going to keep it the same. We're going to keep the F's together and the C's together. They're two different traits. So you have a little f with a big C, and then you're going to have your last F with your last C. Little f, little c. All four of these examples from the top should go down the side also. Let's fill in the boxes. You have this example right here, okay? Big F, big F. So you pull down your F from the top and your F from the side and put them in the box. So you have one capital F and another capital F. Then you have another capital C with a capital C. So you've got big C, big C. That would give you an individual with full lips and cleft chin, both homozygous dominant. So in the second box here, you're going to look straight up, and that's going to give you a big F. And then you're going to straight over to the left. That's going to give you a capital F also. And then here you have a capital C and you have a lowercase c. So you're going to put your capital C and then your lowercase c. That would be your genotype. Your phenotype, or what that would look like, should be full lips and you're going to have a cleft chin. You should be writing them all out, okay, in your own box. So you have a big F on the left here and a little f up top and two big C's. Big C to the left, big C up top. Your phenotype for that individual would again be full lip, cleft chin. I want you to push pause on the video, do the rest of it yourself. I am going to fill all of it in, and then I want you to check your work and make sure you got it correct. But we're going to be doing examples of this in class, and I want you to be able to do it. You should have your genotypes in each box with the F's and the C's together, and you should have underneath it whether they have full or thin lips, cleft or non-cleft chin. And then we're going to talk about the ratios of everything. Okay, at this point, go ahead and take a moment to check your work, and I'm going to clear everything off before we move on. So how many were full lips and cleft chin? I want you to go through and count on your box how many were full lips and cleft chin. Then how many were thin lips with a cleft chin? How many were full lips with a non-cleft chin? And how many were thin lips with a non-cleft chin? If you do the counting properly, you should have a ratio of 9 to 3 to 3 to 1. 9 out of the 16 squares were full cleft, 3 out of 16 were thin with cleft, 3 out of 16 were full lips with non-cleft chin, and 1 out of 16 was thin lips with a non-cleft chin. So we call that the 9 to 3 to 3 to 1 ratio. And that will always be the case if when you have two heterozygous parents for two traits. You do need to remember that. That's the classic ratio for heterozygous dihybrid cross. So dominance relationships, the last slide we just showed only complete dominance, where one allele has full dominance over the other. It's either dominant or it's recessive, but there are a lot of other options than that. One example is that there is incomplete dominance, where neither of the alleles are fully dominant, and instead you'll get a mixed phenotype or a blending of the two traits. There's also codominance, where both alleles show, and one allele typically wins or shows in some cells and not in other cells. The most famous example of codominance is going to be AB blood type, and we'll go through an entire day of blood type. So which one would this be? Complete dominance, incomplete dominance, or codominance? Either attached or detached. That one is complete. You're either dominant or you're not. This one, where it shows red and white making pink, is a mix or blending. That would be incomplete dominance. Right here, where you have a bunch of different eye colors, that is also incomplete dominance, where you have blendings. You have hazel, green, blue, brown, all different kinds of variations in between. And so you have an incomplete dominance. Darker colors tend to show more strongly than lighter colors. This one, where you have a flower that is red and white, that would be an example of codominance.
Sorry guys, you're going to have to bear with me as I do a voiceover on this. The audio is lost on the original recording. So for your example for incomplete dominance, there's an example of some wild cats that come in the colors of blue, red, and purple, and the traits controlled by a single gene with incomplete dominance. So if you have a homozygous big B individual, it's blue, a homozygous little b individual is red, and a heterozygous big B little b individual is purple. So your question is, what would be the phenotype and genotype of the offspring if a blue wildcat were crossed with a red one? In this bottom left-hand square, we are going to do that cross. First, I'm going to place the blue individual across the top with the big B, and then the red one down the side with a little b. Once you fill it all in, you'll find that all of the individual offspring are heterozygous, meaning 100% of them will be purple. When we cross a purple cat and a red cat, can they have blue offspring? And you find, once you fill all that out, that the answer to that question is no. Our next slide is an example of codominance. So instead of having blending, you are going to see both traits. We have a white cow or longhorn, a red longhorn, or a roan longhorn, which is the guy in the middle that has red and white hairs. So your question is, what if you were to cross a roan bull with a red female? So in our example, I'm going to set up a cross with an RW for the roan bull and RR for the red female. The offspring are going to be 50% red and 50% roan. Remember, roan is not red and white make pink. Here with codominance, you see red hairs and white hairs. So this cow or longhorn has two different colors. Our next question is, can roan cows produce more roan cows? So for this second cross, I am going to set it up with RW across the top and RW down the side. What you're going to find is you have 25% red, about 50% roan, and 25% white. So can roan cows produce roan cows? Sure they can. Yes. Have a wonderful day.